Let's move on to 2.3, understanding protein dynamics. As you all know, proteins are dynamic objects. They are surrounded by solvents, and because of interaction with solvents and the temperature, it moves like a live organisms. And this dynamics is often just natural, but sometimes these dynamics can be crucial for their functions. So therefore, understanding protein dynamics is very important. But then issue is that getting the information of protein dynamics from static structures, what I mean is the deposit structures in PDB, is not easy because they are static. And how you can get the dynamics information from PDB largely depends on how the structure was determined. And what that means, the method either is crystal structure or is it NMR or cryo-EM. So in the following sections, we'll discuss how you can get the protein dynamics information from each methods, starting from NMR structure. So NMR is the most natural way of looking protein dynamics from PDB. Obviously, the structures cannot be movie, so you cannot get the movie of protein dynamics, but instead you can get multiple snapshots of proteins in a single NMR structure. So if you open up NMR, it's typically composed of 10 or 20 models, and then they look like this. So you can see some regions are quite converged and other parts are quite dynamic. And you can see that from these overlapped structures that this C-terminus of the protein is quite dynamic, and several loops are a little dynamic, and then other core parts are quite stable and static. And then this information is written in PDB format in model, and model, model 2, and model 3, and model, and so on. So it usually consists of 10 or 20 models in a single PDB, or in MMSIF, there is a model index column at the end, which contains the same information as in PDB format. Cryo-EM has some information, but it depends on how the experimentalists interpret the cryo-EM data. So naturally, the cryo-EM structure consists of many dynamic states of protein. So if you can carefully classify them into different class of dynamic states, like shown here, A to F, then you can make a whole bunch of different static snapshots that represent different states of dynamics. But this is very challenging. And if you open use typical cryo-EM structure, most of them are just contains single snapshot of protein structure, which is just average structures of everything. But recently, some techniques are being developed, and there will be also cryo-EM structures that contains more than single model in a single PDB file. So the way how this works is, as we explained that this cryo-EM 2D image is just a snapshot of frozen protein structures, and then each protein have different poses like this. And if you can carefully classify this, you can group these. Blue ones are having the same shape, which means the same dynamic state, and the others have their own different states. Then you can construct individual 3D image of blue one, green one, and yellow one separately, and then build into separate 3D model structures. That's how you could, in principle, build dynamic, a PDF file containing multiple models representing the dynamics of cryo-EM structure. Crystal structures are more challenging. you can almost say you cannot see dynamics of proteins, dynamics in crystal structures. That's because crystal structures are forced to not have dynamics for building crystals. And because of that, the dynamics is most suppressed in order to get higher resolution. But indeed, if you carefully look at crystal structures, sometimes there's parts that have two alternating states, which is written in PDB format like this with occupancy here. So in crystal structure, 57% has this kind of flipped asparagine here, and then other 43% have asparagine conformation like this. That means that there's some asymmetric part in the crystal structures, but that's usually what you can get most from the crystal structures. But instead, in crystal structures, there's a metric called B factor, which tells you the local irregularity of the crystal structure. So if you look at this structure, then there are blue regions and green regions. Blue regions means they were very regular across different crystals, and crystal asymmetric units. And the green color means that part was especially irregular across different asymmetric units. So you can get kind of idea these green parts were more dynamic than the blue part, so that it was reflected to irregularity in the crystals. So you could easily correlate with the B factor with the protein dynamics, but there's a one caution you have to make, that not all the B factor values are directly correlated to flexibility. So obviously the region having high B factor, which means a very dynamic region, can be translated as dynamic. But then there are also the opposite cases when it is dynamic but having still very low B factor. And that's because there could be extra stabilization at the crystal interface. That means this part was originally quite dynamic, but because of the crystallization, this part was measured as less dynamic. So you have to be careful on interpreting B factor for the flexibility measure of the protein local regions. And actually, at the title of this section, I mentioned either dynamics or uncertainty. So dynamics could sometimes mean uncertainty, and this uncertainty becomes worse and worse if the resolution gets worse. For example, for the same structure, if you draw the electron density at different resolutions, if the resolution is lower than 1 angstrom, then you can see the atomic points, all these nucleus points. But if it gets higher than 3 angstrom, you can barely see where the atoms are located. And if it goes to 5 angstrom, then you cannot even follow where the atoms are. And with this local resolution and dynamics, in practice, many protein structures deposited has modeled regions where there is a low density or overall structure is in a worse resolution. Here's an example. So they determined ligand-containing protein structure, and then I'm showing the electron density of ligand. And based on this ligand electron density, you know there's something there, but you cannot say which direction the ligand should be pointing towards. It could be either this side, or even this side. So how to put the ligand into density is just human modeling. And it's not limited to just ligands, but often it goes to protein side as well. And I'm showing here how the outcome protein structure is built depending on the human modeling effort. This is the same protein which contains somatostatin receptor with the peptide bound. They are determined about the same time, and the resolution is worse at the left part. But then if you carefully look at the atomic structures, the left one has more hydrogen bonds and better packing, no clash. Whereas if you look at the right one, although the electron density was in better resolution, it has lower number of hydrogen bonds, and then they are loosely packed. What that means is that although resolution was the worst on the left-hand side, the modeling was better on it and give you more realistic, physically realistic structure. So this again stresses the importance of human modeling when the resolution is worse, especially when it's worse than 3 angstrom. And in worst case scenario, sometimes backbones could be wrong. This was one of the structures that was deposited into PDB with its PDB ID of 5mm4, and then cryo-EM resolution was 4.5 angstrom. But then people realized this is not physically correct structure, and then people tried to remodel the backbone trace using their software. And then you can see that the B, the right one, has much better hydrogen bonding patterns between backbones, and also better density fit, which means even the backbones could be wrong or incorrectly modeled, or at least it is hard to argue which one is correct than the other. So this kind of error happens somewhat frequently when the resolution is about 5 angstrom EM structures. Let's move on to 2.4, understanding atomic details in PDB. We are keep stressing that we are looking at atomic structures, and the point of looking at atomic structures is to see the atomic details of proteins. But the issue here is that not all the atomic information is correct. In the earlier slides, I mentioned that this happens because proteins are dynamic, and then some parts are experimentally poorly determined. So that part should be modeled by human. And so, but still we learned that protein backbones are generally correct, except for when the resolution was worse than 3 angstrom. So we sort of can trust most of backbone structures for proteins when resolution is better than 3 angstrom, or NMR structures. But there should be really caution needs to be made when you interpret not the backbones, the rest. And here are the items that I especially highlight that you have to pay attention to. First, you have to pay attention to reading hydrogen information from PDB. Second, the water molecules. PDB file has water molecules and their atomic coordinates. And number 3, interpreting side chain atomic positions. So we'll look at one by one in the next slides. So sometimes you'll find PDB files containing hydrogen information there. But then you have to be careful because hydrogens are not something you can easily see from experiments. You can only see at the very few occasions. First is when it's a super high resolution, when it's the X-ray or neutron scattering experiments. I didn't explain about the neutron scattering, but that's something that could be used for structure determination for limited cases. In that case, you can see the hydrogens, locations of hydrogens, and their how much polar those hydrogens are. So if the resolution is super high resolution, you can trust hydrogens. And second case is NMR. You can trust hydrogen positions in NMR because they are subject to NMR spin. And these NMR structures are modeled between the hydrogens that contains either NOE or chemical shifts. So you can trust the hydrogen positions from NMRs because that's a direct measure outcome of the measurements. Still, there are other cases which do not belong to 1 or 2, but containing hydrogens. And you can just think they're modeled. And modeled means that they don't have any experimental support. It's theoretical calculation, and it's up to you to trust or not. Number 2, water molecules. Except for membrane proteins, waters are surrounding all over the proteins. But you cannot easily see that in experimental structures. The reason is because waters are very dynamic. So waters are anywhere, but there could be nowhere in experimental structures. There are some exceptions when you see water molecules, and that's when waters are not dynamic, but ordered by interaction with other molecules. If you look at here, these 3 water molecules are determined in the structure because they are ordered waters, hydrogen bonding to nearby molecules. So they are just stuck there and they have their own density in either crystal structures or EM structures. So if you're lucky, then you'll see these kind of water molecules that are ordered. But if you're unlucky, then you cannot see them, even though they are ordered molecules, or I mean ordered water molecules. Especially that happens when the resolution gets worse and worse. And if the resolution is like 3 angstrom or more, it becomes hard to see water molecule density, even they are ordered. So if you look at carefully here, this is the extracellular region of GPCR. So they should be some water molecules surrounding there. And then if you measure the distance between oxygens, then they are very close to each other, which means that there could be electrostatic repulsion unless some other favorable thing happens there. And I would say this shows the ordered water molecules, although experimentally not deposit there. So if you place the water molecules at the center of this hairpin, then it becomes really a highly favorable structure. So water molecules are not just stabilizing themselves, but it will stabilize this pairing of these 2 parts. But you cannot see that. You can just imagine that. And last part is side chains. And side chains could be easily mismodeled. One reason is because side chains are more dynamic than backbones. So their densities are usually poorer than backbone densities. But besides that, there are certain side chain types that you cannot distinguish from the electron density. What electron density shows is just the shape of the side chains. So it'll tell you what kind of amino acid should come there, but sometimes it doesn't tell which element should come where. If you carefully look at histidines, asparagines, and glutamines, they are kind of symmetric shaped, but their elements are different. So histidines could be in this way, but you can also model in separate way, opposite way. Still, it'll fit perfectly to the density. And same story for asparagines and glutamines that this nitrogen can be flipped with oxygen. And Again, how to fit these elements into density is up to the experimentalist. And when they don't pay attention carefully, they could easily mismodel the positions of nitrogens and oxygens or nitrogens and carbons in this setting case. So when you open crystal structures or EM structures, you have to be really careful on interpreting the side chains of histidines, asparagines, and glutamines. And this is an example that this flipping can happen quite often. This is what is deposited in PDB. And because this ligand part here cannot have hydrogen, this interaction between asparagine and ligand becomes highly electrostatically negative. It's very, very, very poor interaction here. And this is because the structure was modeled incorrectly. This asparagine needs to be flipped in this way so that you can have favorable interaction in two sides. I'm showing you this because it's happening quite often in PDB. Okay, let's summarize the second part of lecture 2. We are looking at protein structures which are outcomes of the experimental determinations. So people try to determine the structures at the condition that is closest to the biological condition, but often there are cases when you cannot do that. So that's why there could be some discrepancy between how the proteins behave in biology and how it is deposited in the PDB. So when you use the PDB file, then you have to be careful and need to understand what kind of modification could have happened in PDB file at the experimental determination stage. And that'll help you to better use PDB structures for many purposes.